Asia, and Dr. Sundaresan, Principal Scientist, CSIR, Central Institute of Medicinal and Aromatic Plants, Research Center, Bengaluru, are with us. We welcome you, sirs, and all the participants for this webinar. Thank you for accepting our invitation, sir. And both of them are eminent personalities in the field of research, both Professor Marimuthu as well as Dr. Sundaresan. And Professor Marimuthu finished his interdisciplinary PhD research degree in zoology and environmental biotechnology at Manonmaniam Sundaranari University, Tamil Nadu. He has published more than 100 publications in international reputed journals and books, book chapters, and so on. And he has been editor-in-chief and editorial board member of several journals. And 2 students got their PhD degree, research degree, and guided 4 master theses. And he has secured research grant of Malaysian ringgit of 8,20,300 rupees, which is in Indian value, rupees value, about 1.5 crores. If I started to narrate his achievements, this 2 hours is not sufficient, so I stop here. And I wholeheartedly welcome Professor Marimuthu sir to deliver his lecture on application of statistics in biological sciences research. Sir, Marimuthu sir, over to you, sir.
Yeah, thank you. One minute. Dr. Stephen, can you see my screen?
Yes, sir. Application of statistics in research.
Yeah.
You can put it in full screen, sir.
Again, sir, full screen.
Yes, sir. Yeah.
This is actually in full screen only.
Yes, sir.
Okay.
Now it's clear, sir.
Okay. Good evening, participants. Dr. Stephen, principal of this St. Joseph College, Bengaluru, and Dr. Joseph, Director for Research, and my colleague, Dr. Sundaresan. Good evening, everyone, and thank you so much for inviting me for this webinar at this evening. And nice to meet all the participants. And thank you for the topic I have chosen for this today webinar is application of statistics in biological sciences research. Right? So without statistics, no research in any fields. So in my presentation, I'm going to cover these are the areas, starting with what is a research. And when you do research, we need to follow some process steps, guidelines to carry out, conduct the research. And when we do research, a lot of motivation behind us, we do follow. And then why do we need to have a statistical knowledge in biological related research? And what do you mean by biostatistics and what are the types? And some of the important terminology related to biostatistics I will cover. And then I will define what you mean by variables. What are the types of variables? What do you mean by data? And what are the different scales of measurement? And there are 2 ways normally we do research. One is observational studies. Another one is experimental research, whereby we do in the laboratory conditions. Observational studies actually is just a field work, and observational, just we observe and we report. And how do we handle those kind of data? And how do we handle those experimental based research? And then how to present a set of data using tables and graph. What are the principles we need to follow while you go for a graphical presentation as well as tabulation. And then a little bit I will touch over on measures of cell tendency and measure of dispersion. And then end with when and how to use some of the basic statistical like t-test, chi-square, correlation, regression, ANOVA, and then end with statistical software used in research data analysis. Okay. So before we move on, and the background slides you see, this is the university where I'm currently working. So since March, I have been stranded here in India due to this pandemic COVID right? And basically our university is a medical university. We do offer medical, dentistry, pharmacy, and biotechnology, engineering, along with business, physiotherapy, nursing. And this is around 400 kilometers away from the Kuala Lumpur International Airport, just 1 hour drive from the Penang International Airport. And majority of 60% of the students are Malaysian Indian students, and around 30% Malaysian Chinese students, and 10% international students. We have a couple of Indian students as well. They are pursuing higher education in our university. What is research actually? It's a systematized effort to gain new knowledge, not research. Research actually systematized effort to gain new knowledge, new method, new development, new device, new protocol. And the protocol may be, minimize the time, minimize the cost, even development of diagnostic, kit comes under development of new diagnostic. Globally, people are looking for vaccine. It's a research, looking for new knowledge, new product, new commodity, right? Yeah. So research actually is a systematic process of collecting, analyzing information. Information means data in order to increase our understanding about the phenomenon, about the biology, about which we are interested in to investigate further. And there are several motivations we do conduct research, and there are several. One is compulsory component. When you go for higher education, master's and PhD, a research component is one of the compulsory component, right? In foreign countries, even master degree itself is a research, it's a kind of mini project. They will submit, there won't be any kind of coursework like Indian universities. India, we do have 1.5 year, we do study about coursework, and one semester we do conduct research. And whereas in majority foreign countries, master degree means it's a research mode. So 2 years project, and they submit a small mini project and submit a mini thesis, and they'll be awarded. So decide to get a research degree along with the consequential benefits. When you are having a PhD or higher education, your salary, your your benefits, your perks, additional benefits higher, and many people they go for higher education, PhD. And then there are several motivation, like there are several challenges we are facing today, unsolved problem, concern over practical problem that initiate the research, looking for.
Sir, you are muted, sir. Marimuthu, sir.
Yeah, it's okay. I have unmuted. Yeah. Can you hear me now?
Thank you, sir. Yes, sir.
Is it audible? My voice is audible?
Yes,
Okay.
sir. Audible, sir.
All right. And doing a research actually is an intellectual joy. Happiness is there, real happiness is there. So first time you got your paper has got published in a quality journal, and first time you got your experimental result as you expected, really the happiness is unmeasurable. And then decide to be a service to the society. Globally, people are looking for biofuels. Globally, people are looking for biodegradable plastics. Globally, people are looking for control of mosquito. These are all related research service to the society. So when you go for research funding, if your project has got any societal merit, a societal value, those particular.
Sir, again you are muted, sir. Marimuthu, sir.
Very often someone is muting me. I didn't do that.
Oh, okay.
Someone is muting. Yeah. So service to the society, looking for biofuel, biodegradable plastics, control of mosquito. So these all global issue, global problems. And if you have anything for this area, any research related to this area, there is a more possibility of getting a project. So there are several motivation conducting a research. So then what do you mean by research process? Research process, usually we do two type of research. One, a supervisor will have a project funded by the different funding agencies like DST, DBT, CSA, ICA, and they will advertise and look for a qualified students to carry out the research. And other way, student will have some basic knowledge, basic idea, and based on his research interest, he look for a supervisor, he look for a university, he look for a research laboratory. With that, he will apply directly to the supervisor and register for a PhD and carry out the research. And whatever the way we do research, first of all, we have to start with the thorough literature review. We have to follow the research process. Thorough literature review means we have to look at what is available so far for the topic we have chosen, the rationale behind what is available, what knowledge is available for the topic you have chosen, and what is lacking. And then we have to, based on that availability of knowledge, we have to formulate our hypothesis, our objective. And based on the hypothesis, based on the objective, you formulate your experimental design. And based on the experimental design, you conduct the study, collect the data, wherein the statistical component comes in, statistical tools comes in, statistical test we need, wherein the student as a scholar, the researcher, we need to have a statistical knowledge. And then using appropriate statistical test, we analyze data, and then we interpret and we report for funding agency, and whereby we also do submit a thesis for a dissertation for a degree, PhD or master's. Okay. So this is a research process thoroughly we need to do. And being a scholar, we have to have the habit of reading a manuscript, very, very important. Literature review, reading a research article will give you a lot of confidence, a lot of writing skills, and a lot of writing the manuscript, and what do you call justification, all the knowledge you definitely will gain. We have to have the good habit of reading a manuscript every day, at least one research article in your field of research. That will help you during your PhD thesis writing. This is my personal experience. So what is statistics? Statistics is nothing but a set of tools, methods used for collecting, organizing, presenting, and analyzing numerical fact or numerical data, numerical observations. Set of tools, methods used for collecting, organizing, presenting, and analyzing numerical facts. A strong background in statistics is essential for researchers in any scientific field. In order to design experiment, to conduct a survey-related research, to analyze the data and present your research finding in a common forum, in a conference, in a public hours examination. So when you go for any research publication, any research presentation in a conference, in a common forum, majority questions during your presentation will be related with your statistical test, will be related to your experimental design. Yeah, majority of the academician, you agree with me? Majority of the question will be posted by the participant or audience will be related with your statistical test. So if you are very good in statistics, you can defend yourself with the full confidence and defend yourself. So why do we need to have a statistical knowledge? To understand the research article thoroughly, the statistical portion. Many scholars will have the habit, when you read the manuscript, research manuscript, title easy, you will understand. Abstract you read, introduction section you read and you understand, and materials and method somehow you will understand. At the end of the materials and method, there will be a last paragraph will be data analysis, and wherein they used to mention what type of statistical test they have used it, and then they use some kind of statistical jargon like t-test, p-value, probability, and wherein majority of the scholars they do get confusion why they have used it, and where without lack of statistical knowledge, they do have a little confusion, and they don't complete reading the full manuscript. Okay, so if you have a good statistical knowledge, definitely you have a you can understand most of the research article thoroughly and the statistical portion of the most scientific article, and then very well you can design your project. You can very well supervise your student. Majority of the supervisor lack of statistical knowledge in supervising the students. Majority of the supervisor in India, I would say. Now, I've been examiner for many Indian universities. I based on my the experience, and I have gone through the PhD thesis, and majority of the thesis, I understood, I found that the statistical portion, majority of the thesis lacking. very weak, I would say. And then we need to know what type of statistical test need to be applied to a particular set of research, particular study. For that, statistics is very, very compulsory component. Maybe you can look at any degree program, any undergraduate or any master degree. Without statistic course of statistics subject, there won't be any degree program. Even business students, engineering students, physio, nursing, pharmacy, biotechnology, medical, all fields, there will be one compulsory paper regarding statistics. Okay. And then learning statistics actually is a very fun, kind of fun. If you love the subject, you will enjoy doing it. Actually, it's a dry subject. If you understand the concept, you will enjoy doing learning statistics. So nowadays, we are very fortunate. We have a lot of statistical tools are available to analyze our data. So if you understand what statistical tests need to be used for a study, for a research, very well you can analyze your data. Okay, a lot of statistical tools are available. Even I would say all the PhD scholar will have a computer at home. Many statistical free softwares are available. Only thing is we need to know how to key in the data and what statistical tests need to be used for a particular study, and system will give you the output very well. You can easily interpret the results, wherein you can go for a quality research publications. And majority of the scholars, I would say in India, Tamil Nadu, South India, due to the lack of statistical knowledge and using inappropriate statistical test, they do go for even quality research they do, but they do go for quality research journals. Less quality research journals, they do go for regional journals, they publish the data due to the lack of statistical knowledge. I hope majority of the academician join this session, you agree with me. Due to the lack of statistical knowledge, they go for regional journals. Even then, the study is valid for, can go for high quality impact journals. Lack of this, they go for a local regional journals. And moreover, Indian University, we have a constraints like, we have to have 2 research paper compulsory when we submit your research project. So very rush, they go for a regional journal with paid journals and a lot of open access journals. They go for it and publish it. Yeah, if someone is having good knowledge, definitely the candidate scholar can go for a quality research journal. So there are 2 main areas of research. One is mathematical research, mathematical statistics. Another one is applied statistics. Mathematical statistics is nothing but a researcher, mathematician, they develop new methods, statistical inference, new methodology, new methods, no statistical test. Whereas in applied statistics, we use those statistical methods for analyzing the data, applied statistics. So biostatistics actually is a branch of applied statistics. We apply statistical method for our data analysis, medical research, biological research, life sciences, arts and science, etc., political science, etc. So statistics used in biological sciences, we call them biostatistics, or otherwise we call them biometry. Statistics used in biological sciences, we call them biostatistics or biometry. Statistics used in all the fields, biology, life sciences, medicine, public health, business, and psychology, etc. Many fields. I would say without statistics, no research in any fields. And then little bit I'll cover some of the important terminology like population, sample, parameter, statistic. Population means complete collection of all elements, like to study about the diabetic prevalence in India, a prevalence of COVID in Tamil Nadu, prevalence of COVID in Bengaluru. It's about population. To study about the population, we do not have sufficient time. We do go for a sample. Sample means a portion of the population selected for the study, to study about the population. Okay. So conducting a research in a population is very, very tedious and time consuming process. For that reason, we do go for a research using sample. For example, birth weight of 100 baby in a particular hospital, birth weight of 100 a patient in a particular hospital, particular city, right? So population sample. Population means whole. Sample means a portion of the population used for study. Then what do you mean by parameter and what do you mean by statistic? Numerical measure of a numerical characteristics of a population is called parameter. Similarly, the numerical measure or numerical characteristics of a sample is called statistic. So the terminology we need to remember: parameter and statistic. Numerical characteristic like mean, median, standard deviation of a population, we call them parameter. Sample standard deviation, sample variance, coefficient of variation, mean, for sample characteristics, we call them statistic. Then what do you mean by variable? Variable is nothing but whatever we measure, property, attribute. For example, when you conduct a study, survey related research, wherein you include a question like gender, age, family income, what is her age, and what is her height, what is her weight, and each one actually is attribute. Each one actually is a property of the subject. We call them as a variable. Blood group, gender, body weight, height of a patient, etc. Or you study about the chlorophyll content or total phenolic, or total flavonoids are examples of variable. Then what do you mean by data? Data is nothing but whatever your observation, measurements, gender responses, survey responses, etc. Data comes in two forms, one in qualitative form, another one is in quantitative form. Qualitative form actually is a gender. We look at the quality of the subject. Qualitative data or qualitative variable. Gender, smoking status, marital status. Have you got married? Have you got, do you smoke? What is your gender? These all quality of the subject. We call them as a qualitative data. And there are some data, we call them as a quantitative, meaning to say that measurement of chlorophyll content, measurement of flavonoids, a measurement of alkaloids, phenolic content, we call them as a quantitative. Quantitative, we use some kind of device. We use some instrument to measure. For example, your height and weight, your blood glucose, right? Your cholesterol measurement, we use some kind of instrument to measure. We call them quantitative data. So variables, there are two types. One is a qualitative, another one is a quantitative. Qualitative, we can call them as a categorical, and quantitative, we can call them as a numerical, right? For example, qualitative or categorical variable, qualitative data, marital status, and what is your race, what is your gender. It says that it's a defined category. We call them as, this kind of variable just explain about the quality of the subject. Okay, qualitative, categorical. Then quantitative, there are two types, discrete and continuous. Discrete actually is a whole number without any decimal values. Number of children in a family, number of text messages you have received since morning, number of seeds produced in a plant, number of fruit produced by a plant. We call them as a discrete, wherein we use the whole number. A number of bacterial colony in a petri plate is a discrete. Any family, do we tell them that my family has got 4.5 children? No. Wherein we use a whole number, right? So whole number is a discrete. What do you mean by continuous then? Continuous actually is a measurement process wherein we use some device to measure them, and then we'll have a decimal values. Your height, your weight, electricity, your cholesterol, your hemoglobin, what we call plant characteristics like phytochemical components like flavonoids, tannins, saponins, and antioxidant alkaloids. We call them as a continuous variable, meaning to say that it comes with a decimal value. Do you think exactly your body weight will be 70 kg or 80 kg? No, right? Your body weight can be 70 kg, 50, 500 grams, and milligrams, and etc., microgram, etc. There won't be exactly. So another example for categorical nominal variables, blood types, gender, which race you belong to, Malay, Chinese, Indian, and what is your favorite color, and what is your favorite actor, type of car you prefer, what type of media you prefer, are examples of categorical variable. And another variable we call them is called ordinal variable. Ordinal is a ranked variable, right? When you go to hospital, doctor will ask you what type of, how do you feel about your pain? Mild pain, moderate pain, or severe pain? And quality of the meet, quality of the program, quality of the webinar, how do you rate 1 to 5? How do you rate the quality of the webinar organized by this organizing committee, 1 to 5. We call them as a ordinal variable. Okay, and then quantitative variable. Quantitative variable is a measurement data. On quantitative variable, convey information regarding the amount data that is given numerically. Height, weight, your age, hemoglobin are examples of quantitative variable. And then we also need to know independent and dependent variable. While conducting a experiment at laboratory condition, we do deal with two type of variables. One is independent variable, another one is a dependent variable. As a researcher, as a scholar, we do manipulate the independent variable, controlled variable, experimental variable. Whereas in the dependent variable or the effect of independent variable, dependent variable are the measured variable, dependent variable are the outcome variable or response variable, dependent variable are the functions of independent variables. Okay. For example, you study effect of temperature on the hatching rate of fish eggs. So what is independent variable? Temperature here is an independent variable, and the outcome variable, response variable, hatching rate is the dependent variable. All right. Yeah. So there are two types of data. How do we differentiate? Discrete and continuous. Discrete actually is a whole number. Continuous actually comes with the decimal values. Continuous, it comes with decimal values. So discrete actually, easiest way you can remember, counting process involved. Number of text messages since morning you have received. Number of bacterial colony in a petri plate. Number of children in a family. Number of phone calls you have received since morning. It's a kind of discrete whole number we report, we record, right? And the research question will be how many colonies are there, how many eggs produced, and how many children you have, how many bacterial colony, how many phone calls you have received, how many text messages you have received. So these are all called discrete data, discrete variable or discrete data. Counting process involved, and the research question will be how many, starting with how many. Is just a way you can remember. Then what do you mean by continuous? Continuous actually comes with the decimal values, measurement process involved, and the research question will be how much milk, how much body weight, how much blood glucose, how much your hemoglobin, how much the body weight, how much phenolic content, flavonoids content, etc. Cholesterol, albumin, globulin, etc. Right? Without gaps, this value we do present. So now, one or two participants, you can unmute and tell me whether this data is a discrete or continuous. Voltage of electricity in a power line. Any one of the participants, you can unmute and tell me. Is there any option? The host, is there any option? The participant can unmute and tell me, respond to understand. Tell me voltage of electricity in a power line is a continuous or discrete. Whether it's a measurement process or a counting process. The measurement process will be continuous. Tell me number of eggs that hens lay.
Discrete.
It's a continuous, discrete. Time it takes for a student to complete a particular exam. Time continuous time. It comes with minutes, seconds, milliseconds. Income
Discrete.
Discrete sometimes after tax deduction, sometimes we call them treat them as a continuous, sometimes only decimal values. So continuous. Family annual income, continuous. Number of colonies in a petri plate, whole number, we use them, is a discrete. Yeah. Suppose you are measuring a OD of a solution using a spectrophotometer reading.
Continuous.
Fantastic. So if you understand this basic, why we need to understand this, handling with the discrete data, handling with the continuous data, that is a different statistical method. When you read textbook, they don't cover this much detail. So when you understand this will dictate us what statistical tests need to be used for the particular scenario. Okay, so far we have discussed the type of statistics, variables, types of data, etc. And then further defining the data, there are 4 scales of measurements have been commonly used. They are called nominal, ordinal, interval, and ratio. So very, very important. These 4 scales of measurement will help us to choose appropriate statistical test for analyzing our data. If you understand this 4 terminology, will give you some basic prelude, some idea to choose appropriate statistical, narrow down the statistical method to go for data analysis. Nominal, I have already explained, is called the qualitative categorical data. Nominal, qualitative categorical data. For example, gender, marital status, yes or no, right? Do you have diabetic, yes or no? Are you hypertension, yes or no? And what is your profession? These all questions looking at the quality of the subject. We call them nominal scales of measurement. And next one is called ordinal measurement. Ordinal means is a ranked variable, ranked data. For example, students' grade, we record them. A grade, B grade, C grade, D grade, and failure. When you go to hospital, pain rating 1 to 5. High, 1 to 5. Strongly agree to strongly disagree. It's another way of, Likert scale, ordinal scale. How do you rate the quality of your, how do you rate your satisfaction? Very satisfied to very dissatisfied, neutral. This is all called ordinal variable, ordinal data, ordinal variables. And what do you mean by interval? Interval actually is there is no any zero, there is no any real zero. There is no any real zero. Even zero also you feel that feel the property. Even zero also you feel the property. For example, temperature. 0 degree Celsius also we feel the temperature. Negative degree Celsius also we feel the temperature. Okay, so there will be equal differences, but we do have the real property of the zero. We do have even negative value also we feel the property of the thing, a property of the subject. And what do you mean by ratio then? The ratio, remember, starting value is a 0, ending with infinite. Starting is a 0. If, for example, you do not have money in your wallet, you feel the nothingness. If you don't work, you will get 0 salary. You feel the nothingness. If you don't have money in your wallet, you feel the nothingness. Okay, whatever the variable, starting with the 0, if you feel the 0 means nothingness, we call them ratio scale. So if you understand this 4 terminology very well, you can narrow down your statistical test. Remember, nominal, ordinal scales are called as a parametric data. Interval and ratio scales are called as a parametric data. So you are dealing with nominal, ordinal data, you have to go for non-parametric statistical test. If you are dealing with interval and ratio scales of data, you have to go for a parametric statistical test. So there are 2 types of statistical tests, parametric and non-parametric. Parametric data, usually interval scales and ratio scale, provided the data should follow normal distribution. Provided the data should follow some of the assumption, and we can go for a parametric. If not, we have to go for a non-parametric statistical test. Okay. Yeah, nominal, ordinal. Ordinal means grade of a student, socioeconomic status, Likert scale, strongly agree to strongly disagree. We call them as a ordinal. Interval, example, temperature. Sometimes, when you go for competitive examination, there will be a negative marking. When we score, if every wrong answer, if you give them some negative marking, sometimes some students will get 0 marks, some students will get negative mark. Those conditions, we call them as a score, we call them as a interval. Okay. And ratio means your test score, your income, rainfall, phenolic content, phytochemical components, soil qualities like minerals, heavy metals, quantification, rainfall, price of a commodity. We call them as a ratio scales of measurement. So now you tell me eye color. You are recording the eye color. Tell me this is a nominal, ordinal, interval, or ratio. Could you please unmute and tell me? I will give you 30 seconds. Eye color to test the understanding about the scales of measurement. Eye color actually is a blue, brown, green. It's a qualitative. We call them as a nominal. Rating scale, poor, good, and excellent. Ordinal. Fantastic. Exam score as it is, your minimum mark is 0, maximum is 100. Minimum is 0, There is no any negative marking.
Ratio.
Ratio, fantastic. How about salary? How about salary? Is it ratio or interval? Salary, is it what type of scale?
Interval.
No, ma'am.
Ratio.
Ratio. Age starting is a 0. Age actually is a ratio. Ranking of a football team in India. Ranking of a football team or cricket team in India. Ranking.
Ordinal.
Ordinal. Right. Nationality, what is your nationality?
Nominal.
Nominal. Temperature.
Interval.
Temperature.
Interval.
1 minute. Sorry for it. Hello. Yes. Yes, ma'am.
Ratio.
Just wait 1 second, participants.
Sorry for it. I had a important call. Okay, right. So temperature is an interval. Postcode, every city we have a postcode, right? What type of scales are measurement? Postcode.
Ratio.
Is it ratio? Postcode for classification. Nominal. Doesn't mean that this particular postcode is better or inferior or superior. No such. For a classification purpose, a number has been given. That's it. Postcode actually is a nominal. Your body weight, your blood glucose, your hemoglobin. Tell me. Body weight, blood glucose.
Body weight ratio.
Blood glucose. Anyone will have a negative glucose, negative hemoglobin. No such, right? So we call them as a ratio scale. All right. So there are two types. Yeah, fantastic. So far you have understood the types of scales of measurement, types of data. And there are two types of research normally we carry out. One is observational research. Another one is experimental research. Observational research, we don't manipulate anything. We go for a field visit. You go for a survey-related research. It comes under observation. We don't manipulate the subject. As it is, you record, you collect the data, you report. Whereas in the experimental research, we do manipulate, we do control some variables and measure the outcome variable, observe the response variable. We deliberately control them in a laboratory condition. We call them as a experiment. Okay. And then statistical method, there are two types. One is a descriptive statistics, another one is inferential statistics. Descriptive statistics is meant for summarizing a set of data, set of observations, summarizing it like a mean, median, mode. It comes under descriptive statistics. And then what do you mean by inferential statistics? Inferential statistics is nothing but comparing or inferring. To study about the population, we do go for a sample. So using sample data, we infer about population, inferential. The term itself has got the meaning. So descriptive statistics, we use mean, median, mode, standard deviation. And then sometimes we visualize the data using a graphical illustration, histogram, frequency, scatter plot. We call them descriptive statistics. Inferential statistics tries to infer information about a population by using using sampling, using sample data to study about the population, to compare the differences and predict about the larger population. For example, asking 1,000 individual, estimating to predicting who will be the next Prime Minister of India. This is called inferential statistics. Asking 1,000 people or asking 10,000 people in different cities to estimating who will be the next Prime Minister. approximately we can estimate or anticipate who will be the next prime minister. This is called inferential statistics, having small group of data to infer about larger opinion about the population. Okay. So when you have a tabulation and graphical illustration, we need to follow some kind of cardinal rules. Graph actually is a mathematical picture, right? And graphical presentation very easy, very convenient to convince any audience. Even normal layman also you can make them understood about the data. So when you go for tabulation or graphical illustration, we have to follow rigorous what you call cardinal rules. So every table will have a clear title. The title should reflect what content we have given in the table. Every table should have a clear number, distinct number. The number should be reflected in the text as well as in the table. There will be a column heading and row heading, and abbreviation need to be avoided as much as possible. If no other way, you have to include as a footnote, you have to give the expansion of the abbreviation, like a foot conversion ratio, specific growth rate, whatever it is. And then unit of measurement, very, very important. Even PhD scholars also sometimes they do mistake. They will miss what the unit of expression, height, weight, phenolic content, mg per liter, microgram per liter, gram or milligram. If you don't give the unit of expression, so audience will have different outlook regarding your data, right? And sometimes we use someone's data for your thesis or a paper. We have to cite them. If you don't cite them, it is called plagiarism, right? So we have to cite them in the methodology. The particular table been adapted from so and so author, so and so journal, etc. We have to cite. So this is actually a kind of model table. Look at here. Table number is there, title is there, and then what data means. They have given mean and standard error of mean. They have also mentioned the detailed explanation for the data, what information they have given in the particular table, and then they have also mentioned the sample size. If you don't specify this unit, for example, look at the unit of presentation, international unit per mg per protein. And they have a freshwater fish, they have a seawater fish, and then variety of fish species they have studied. Actually, it's a huge data, a huge 1 year data they have compiled in one simple graph, simple table. So this is the way you have to go for it. Look at here, another study. So they have used abbreviation, unit is present, abbreviation expanded as a footnote. We have to go for it. So minimal bactericidal concentration. Actually, when you go for a thesis, when you go for a research, it's a kind of document. But document should not be any mistake or any error. Document should be very clean and pakka, right? Yeah, abbreviation need to be avoided. If no other way, you can expand and you can use it. And this is the way normally go for a graphical illustration. Title, most of the journal table will be at the top, whereas a graphical illustration at the bottom. But no such idea why they have used it. But majority of the journals, no scientific reason, but majority of the journals we do follow this policy. So measures of central tendency, measure of dispersion, mean, median, mode, it comes under measure of central tendency. Standard deviation, range, variance, we call them as a measure of dispersion. So descriptive statistics and the graphical illustration can be used for a particular data. Let's say you are dealing with the categorical data, you can go for a frequency, you can go for a percentage. If you are dealing with the continuous data for measure of location, you can go for mean, or measure of central tendency, you can use mean and median. Suppose you are going for continuous data, measure of variation, a dispersion, you can use standard deviation, or you can use range, minimum, maximum, etc. For graphical illustration, categorical data, there is a different chart facility or graphical tool, you can use it. For presentation of continuous data, there is a different method, statistical test, statistical graphical method can be used. Categorical data, you can use bar chart or pie chart. Whereas continuous data, you can use histogram, you can use a box and whisker plot, you can use a dot plot, you can use the scatter plot. Okay, so remember what type of data we are dealing with. Accordingly, you have to choose even a graphical illustration. Okay, and then different statistical measure, like mean, median, mode, frequency, percentage. For example, you are reporting gender, reporting married and unmarried or not married. You go for a frequency, you present them. This much percentage of the participant enrolled in this session are male. This much percentage of the participant enrolled in this session are female. Okay, so categorical data. And this much percentage of the participant have got married, or this much percentage of the participant how they are academician, and this much percentage of the participants are what you call research students. So if you measure them height and weight of the participant, we go for mean, median, or dispersion, we go for standard deviation, we go for minimum age and maximum age, etc. So there are two types of statistical test, parametric test and non-parametric test. Parametric is it looks for interval data and ratio data, provided it requires some assumption, some requirements. The data is supposed to follow some normal distribution. There should not be any outlier, right? So we can go for a parametric statistical test. Non-parametric means it doesn't matter, any type of data can be used. Suppose you have ordinal data, nominal data, interval data, ratio data. If the data does not follow normal distribution, like a bell-shaped distribution, we go for non-parametric statistical test. Okay. I think I have running out of time. So this, if you understand this particular table, is enough for this session. There are different statistical tests are available. Very commonly used statistical test in biology. One sample t-test, two sample independent t-test, paired t-test, Pearson correlation, one-way ANOVA and two-way ANOVA. So these columns are called parametric statistical test. And this column are equivalent non-parametric test. Remember, each parametric statistical test procedure, there is an alternate statistical test procedure in non-parametric. For example, one sample t-test comparing with the gold standard. For example, you are purchasing a product from a supermarket. Purchasing any product means that milk or any kind of a commodity product you're purchasing. Do you think there will be a net label 100 gram or 200 gram or 1 kg? Or regularly you are purchasing a sugar from a supermarket. Do you think exactly 1 kg will be there? The measurement? Do you think exactly 1 kg will be there? Definitely no. Definitely no. So how much can be allowed? 95% of the time, if you get exactly 1 kg as a customer, we are happy with. 95% of the time, when you do sample, you quantify. When you get 1 kg, they mention. If you get 1 kg, is well and good. 5 times if they do mistake, 5 times if there is any error in biological sciences, acceptable. 5 times the 1 kg is very plus or minus, acceptable. Okay. 95 times if you are meeting the 1 kg as a customer, we are okay with. So those kind of study, those kind of data, you can compare with one sample t-test. Independent sample t-test comparing with the two independent groups. Let's say I have two independent group, male and female. I do study about, or rather I have two types of plant A and plant B. Species A, species B, I will quantify the chlorophyll content. So which I do 10 samples from species A and 10 samples from species B. So comparing the chlorophyll content between the two plant species is called independent sample t-test, provided the data is supposed to follow normal distribution. If not, I have to go for a Mann-Whitney U-test, non-parametric. Paired t-test, paired means before and after. Same subject, we measure them before and after. Let's say you have 100 diabetic patients and you have some herbal supplement for controlling the diabetic, right? And how does a herbal supplement control the glucose? Before the administration of the drug, you measure the glucose content. And after having the 2 months of the drug administration, you measure them, again, the glucose content of the 100 patient of diabetic patients. So before and after, is there any differences? We call them paired t-test. Suppose you have a height and weight of 100 participants. Height and weight, are they related? Height and weight or number of years of experience in teaching and their annual income. Is there any relationship? Or people working in a private sector, number of years of experience and their income. Is there any relationship? We go for a Pearson correlation, provided the data is supposed to follow normal distribution. If not, we go for a non-parametric Spearman rank. The purpose of the test to assess, is there any relationship? or you have 100 students, university students. We are measuring the height and weight. The height increases, weight is increasing or not. Is there any relationship? Or number of hours the student they study for exam and their final exam marks. Is there any relationship? Or number of hours you do exercise and your weight loss. Is there any relationship? More the hour you do exercise, is there any more the body weight loss? We go for a Pearson correlation. Suppose when we can use it one way ANOVA. If you're having more than 2 independent groups, comparing comparing chlorophyll content of 3 different plant species, or cultivating a plant in fertilized with a different fertilizer. Productivity or measuring the wheat or productivity of a particular plant species, 3 different fertilizer, which fertilizer is good, you go for one way ANOVA. In non-parametric, we go for a Pascal-Wallis. Another test is called, very commonly used test is called two-way ANOVA. Two-way ANOVA means you have a 2 independent variable, 1 dependent variable. We go for a two-way ANOVA. And equivalently in non-parametric, we have a Friedman two-way analysis of variance. Okay. So nowadays we have a lot of statistical tools, very fortunate, even all the scholars, academician, some are paid, some are free. I would say Microsoft Excel is a very fantastic tool for learning statistics. And another software is called SPSS. And this is actually a paid version, paid one, and very fantastic, very easy to learn statistical tool. Being a scholar, being a researcher, if you are very good in any one of the statistical tool, that will serve your purpose. Fantastic. Definitely you can do quality research and quality research publication. Okay. So name a few statistical software like SPSS, PSPP, SAS, Data, RStudio, RStatistics, MATLAB, Rapid Prism, Minitab, PAST. There are many more, many more. You can use it any one particular statistical tool for data analysis. Okay. So I have come to the end of the session, my talk. I've been providing data analysis services and consultation for PhD thesis work. Any PhD scholar, PhD student, or any researcher for the project work. If you're looking for data analysis services, you can contact me to this email or to this WhatsApp number. You can contact me anytime. And later I will share these slides to Dr. Stephen and also Dr. Sundaresan, my colleague, my friend, my brother. Okay. So if you are looking for any data analysis services, you are most welcome. You can reach me. You can contact me through this email. And tomorrow onwards, I'm conducting a 3-day workshop on data analysis using SPSS, how to use this particular tool for data analysis. And the registration still open until tomorrow 10 o'clock. Seats are available. Plenty of seats are available. If you are interested, any one of the participants, any one of the participants are interested, you can contact me through the email or through WhatsApp number I have given you earlier. So I'll give you the brochure. Okay. And meanwhile, I will ask Dr. Stephen to share this brochure to all the participants. Okay. So with this, a good opportunity, I thank Dr. Stephen and the management for this St. Joseph College, Bangalore, and the principal of this college and director of this, director of research for the St. Joseph Research Center, and Dr. Stephen and Dr. Sundaresan for giving this good opportunity. Thank you so much. So if you have any questions, I'll be happy to answer. I think I have rushed. Yeah, thank you. I think I have taken more time.
Yeah, for your enlightening lecture on research methodology, especially the statistical part, especially in the biosciences. You have shown a clear-cut path for the early career researchers, what to do and how to do research and so on. And thank you for your valuable time, and you readily accepted our invitation to be a resource person of our research webinar. Now the session is open for question and answer. You can use the chat box for your questions, or you can directly raise your hand so that we'll unmute yourself. You can ask the question directly to Professor Marimuthu. There is one question, sir. I'll read out the question in the chat box.
Yeah, please, sir. Yeah.
For the faster completion of the PhD, is early publishing the research papers on the journals is only the goal for the researchers, or to conduct the research by the aims and objectives slowly? What
Could you
is
please read
your
out? Could you please spell out, sir, your question? I couldn't.
yeah, is for the faster completion of the PhD research, is early publishing the research papers on the journals is the only goal for the researchers, or to conduct the research by the aims and objectives slowly?
Actually, when you do research, if you have one or two quality research publication for any PhD is well and good. Early you publish, well and good. Early you do the research. Reason, many people they look for the same topic. For example, by the time you complete your PhD, if you don't publish it, you wait for, and someone else will publish it, the same topic. So as you get the results, you go for a quality research, you go for a publication. Don't leave any data. Even sometimes you have to go for even negative data, negative results also, you can go for a publication. Sometimes you are looking for some property of a plant. If the particular plant doesn't show any activity or any kind of antimicrobial activity, don't leave it. In negative results, you got it. Nothing wrong in that. Some journals, they do accept negative results. So boldly, this is what I have done. You have to go for a publications. So at least you have to have a 2 quality paper for a PhD. Only you publish is well and good.
Thank you, sir.
Yeah.
If you have any questions, dear participants, you can post it in the chat box, or you can directly ask Professor Marimuthu.
Any questions?
I hope the message is conveyed loud and clear. So, on behalf of the St. Joseph's Research Center and St. Joseph's College, I would like to extend heartfelt thanks to Professor Marimuthu for readily accepting our invitation and being our resource person. And already in the chat box, many participants expressed their view about this webinar, and it is useful for many people. And he is also conducting sessions already as posted in his presentations on statistics using SPSS and also MS Excel. And those who are interested can reach Professor Marimuthu through his email ID gncexcel@gmail.com, which will be posted later in the chat box. You can reach him, and he is a down-to-earth person. You can get his help at any time. Okay. You can contact him. Thank you, sir. Thank you so much.
Yeah. Thank you, sir. Thank you so much. Thank you.
Now, thank you, sir.
Thank you.
Now we'll move on to the second session. With us, Dr. Sundaresan, Principal Scientist, CSIR, Central Institute of Medicinal and Aromatic Plants Research Center, Bangalore. Dr. Sundaresan finished his PhD research degree on taxonomic studies on the flora of Tirunelveli Hills at St. Xavier's College, Palayamkottai, and he has developed 7 different varieties of various medicinal plants and have a patent on SCAR, SEA, or marker on Philanthus species, an hepatoprotective plant species. He has nearly hundreds of publications in international journals, books, and book chapters, conference proceedings, and so on. He is not only expert in classical taxonomy, but also well versed with molecular characterization of germplasm collected from Western Ghats and the Himalayan region. And today, we will be talking about the recent trends in plant science research, and career prospects in CSIR, Council of Scientific and Industrial Research. On behalf of St. Joseph's Research Center at St. Joseph's College, I would like to invite Dr. Sundaresan to deliver his talk on recent trends in plant science. Sir, floor is now open to you. You can start, sir.
Thank you, Dr. Stephen.
Yes, sir.
Are you able to see my slides?
Yes, sir. Yes, sir.
It's fully visible, right?
Yes, sir. It's clear, sir.
Okay. Audio is clear?
Yes, sir. Audio is clear. Loud and clear, sir.
So very happy good evening to you all. You had listened a very good lecture by my brother, my colleague, Dr. Marimuthu, a very good biostatistics scientist. And we are working together for a long time, I think more than 20 years. So I brought him because many of the scholars, early career scientists, it will be useful, this lecture maybe, I think. And here I am in CSIR CMAP, Central Institute of Medicine and Aromatic Plant. It is a regional center. We have the head office in Lucknow, and we have 4 centers, one in Hyderabad, one in Bangalore, and 2 in Uttarakhand. This topic I have selected, actually I wanted to give a very focused research finding from our lab. But Dr. Stephen told it should be a little bit broad so that the scholars or students may get benefited. So I have chosen this topic because this is very important. What was our past and what we are doing and what is our future in research. So with that aim, I have given this title, that is a recent trends in plant science and career aspects in CSIR, because it is very important as a research students. You must know, after completing your MSc or MTech or PhD, where to go and what are the research prospects are available, especially uh, especially for the biology students, because many of the students they have a question where to go. Okay, we have completed the PhD. So if you have a clarity before enter into your career, then it will be very much helpful for you. So with that objective, we have taken one more half of the part of that presentation will be career aspect in CSIR. So with a small introduction, I think I can start my talk, the recent trends in plant science. So I have put this picture. It's a small cotyledon purposefully. It's not a screen saver, or unknowingly I have not put actually. It has been displayed with a purpose actually. So whenever you are seeing a big thing, actually, you should not forget your history, or you should not forget your forefathers. Like that, I have put the small cotyledon. You can see the cotyledon, and later, last in my lecture, I will explain why I have put this slide actually. Okay. So every seed grows into something amazing. We don't know what seed has what capacity. We don't know. So we should not underestimate any science or anybody, even any science. Sometimes people used to tell you are doing some very fascinating science, or we are somebody is doing some taxonomy, some morphology work. It's not like that. Every science has its own benefit and its own strength actually. So every seed, you don't know what you will find, what you will invent, or what you will reach actually. So everybody should proud of your own science, whether you are a zoology, botany, within botany, physiology, morphology, anatomy, biotechnology, phytochemistry, etc., etc. So you just see this cotyledon. Later I will explain why I have put this actually. Okay. So whenever we talk about recent trends, anything recent trends, we should not forget about our past actually. That is very important. Recent trends, everyone know what is going on, what may be the future. These things we can predict your future, and you can visible your present. But you, many of you may not know the past actually. If I ask you, any of you, the scholars, what is the best invention and best finding till date in science actually? Suppose if I ask, what is the best invention, or what is the best science till date actually? Till date, any answer? Anybody can give the answer. What is the best invention? Which is the best invention, and which is the best finding in this science till date actually? Can you, anyone can give answer?
Vaccine.
Till date. Vaccine is the future. You are talking about like COVID vaccine, something like that, right? I'm asking till date.
No, sir. The vaccine that's already come in.
Already come. That is the best research you are thinking actually. Okay, so you are in present, okay, or little past, but I am asking the best one. You just go through this slide actually. According to me, these two invention and these two science is the best research finding in till date. Why? I will come to know later. Okay, the first one, if you see, it is a very simple microscope. This was invented by Anton Van. I think many of the microbiology students might be knowing. And the second one is the Robert Hooke's. These are two inventions. Both are very simple microscopes. And the first one, Anton found a small animalcules. That is, later it is called as a microbes. And the second one, if you see, he just looked a cork actually. And cork, and he called it's like a room, and so that he called it as a cell. And that time he did not know he had discovered the plant cells actually. So according to many of the scientists, it is not only my opinion. If you see, till date, this is the biggest, the very big invention and finding. It is in the 16th. You just imagine, it is 16th century. It is not in the latest century. So these two invention, the basic microscopes, these were the very, very basic and very main invention. And the findings, right side, the finding of microbe and the finding of plant cell. These two are major invention and finding till days in the science. So how far have we come since the day from Anton Van? So if you see that today we have plenty very sophisticated microscopes, SEM, TEM, confocal, fluorescent, etc., etc. You can see a pollen, you can see a trichome which can produce the leaf, oil, and even you can see the moving cells actually moving the organelles within the cell. And another one is the stamens actually. You can magnify and you can find out n number of findings. So when you just imagine from here to reach, it took how many years actually for us. So with these helpful things, how much achievement or how much milestone we have achieved. I have put few major milestones. These all are you know very well, but I just want to remind all these things. The one is the Carl Linnaeus systems of naming the plant, naming the organisms. Another one is Darwin's theory of evolution. That is very important finding. And Mendel's law, that is the genes and inheritance theory. That is a very, very important rules of heredity. And another one is DNA double helix model. That is very, very important milestone. And next one, the regenerative, that is the somatic cell. That is a plant tissue culture, invention of plant tissue culture. So if you see, these are few major milestone in plant science, and that automatically lead to many Nobel prizes actually. Here, n number of Nobel prizes has been given. I have enumerated just 4-5 important Nobel prizes, especially for the pigment chlorophyll and especially for the alkaloids finding. Because you just imagine 1947, the Nobel Prize is awarded for alkaloid. With the all your medicines, most of the medicines, either it may be for a diabetic, it may be for a cancer, it may be a high blood pressure, any non-communicable diseases or communicable diseases, mostly they all are from secondary metabolites. These secondary metabolites are major groups are from alkaloid. And the green revolution, so it is very important Nobel Prize to Norman Borlaug actually. And anti-malarial drug from Artemisia, that is it is a very big and finding, and this got Nobel Prize in 2015. And the latest one for plant science, that is the CRISPR-Cas9 genetic diseases. I think you might all know actually. So with this small introduction, so I have given a small glimpses actually. I just will take you people to this 6 major topic. Don't worry, I won't take much time. Just I want to go through very quickly where we were, where we are, and what will be the future. If these all are published data, there is nothing novel, but you can feel, you can realize what is the importance and which area we can select for your PhD or your research or your future career work actually. Because these are very important 6 areas. I have given these 6 areas. Most of the plant biologists are working within this only. Either it is a crop improvement, in vitro, biofactory, drug discovery, plant authentication, or omics actually. Or omics, synthetic biology will come inside omics only. So why crop improvement I have put first actually? India like country, you know very well, India like country, so much population. The population, I think I don't need to say. So we have to feed billions of population, the people. So it is very improvement. The first foremost technology we require is crop improvement technique, because we cannot import our food crop or our fruit crop or anything actually. So it is very important, our crop improvement techniques actually. So whenever you choose your career in any of these six actually, I definitely, we have a bright future. N number of vacancies are available, n number of research opportunities are in these, among these six topics actually. So that carefully I have selected. The first one is the crop improvement technique. As I told, these four techniques, very common techniques, till date we are following. We started with the cross breeding actually. So cross breeding, I think we know, you know very well, the combining two sexually compatible species to create a variety, that's all, with of course with the desired trait of the parents. So this we are doing till date. Parallelly we are doing, we are not leave this, because this is the base for each and every breeding technology, whether it can be a mutabreeding or polyploidy or protoplast fusion. Without this knowledge, crossbreeding technique, we not achieve all these things. So crossbreeding in all crop it is possible, and this is a very oldest technology. The only small disadvantage is it takes time. It takes not time, it takes years to complete, to bring with a variety. So you have to go for lot of crossings. You need lot of crossings, back crossings. You have to wait for till the desirable try to get. So it takes lot of time. Then we focused mutagenesis actually. Mutagenesis using of chemical mutagens or rays, gamma rays, beta, anything rays, radiation therapy. So we are selecting the traits, new traits. So it is called mutagenesis. N number of works are going on in food crops, fruit crops, medicinal crops, etc., many crops. And polyploidy, that is a wonderful technology. In mid science, they have developed the multiplication of number of chromosomes in a crop to impact its fertility. You may not know about the impact of polyploidy. Many of the fruit, whatever we are eating, many of the vegetable, whatever we are using, they all are the result of polyploidy. And the latest one is the protoplast fusion of cells or cell component to transfer trait between two species, that is the protoplast fusion. This is a very attracted in 90s and 20s. Many people, even still, most of the vegetative crop, they are using this technology. So this is the past actually. Then middle year, we started to work with QTL mapping, quantitative trait locus mapping. So what we did actually, we have completed the mapping the population of a particular crop or a fruit or medicinal plant, anything. And then we started, which is the desirable trait actually. So QTL mapping is a very good technology. Still many of the food crop, agricultural traditional crop people are using. That is called, again, that is called marker assisted breeding. And another one is the QTL pyramiding. That is again a very good searching of a different beneficial loci. Many of the soybean mosaic virus resistant genes, they have got it from this technology only. And genetic engineering and transgenic pyramiding, this is the earliest technology what we are using till date. N number of drought tolerant, n number of disease resistant varieties, especially in salt and drought tolerant wheat and rice. This has been developed through this technology. So what is one example I can give? A non-narcotic poppy. Poppy, you may know, it is a Papaver somniferum. It is very important narcotic crop. It is banned for open cultivation. If you want to cultivate, you have to get permission, approval from higher authorities, or some authorities are there for that, narcotic control, narcotic bureau. But this opium poppy, it is a source of very dangerous narcotic drug, that is morphine and heroin. Okay, so that's the reason it is restricted. But it also has the morphine actually. That is a very good anesthetic compound of alkaloid actually. So but how we can develop a variety without heroin or without morphine. There is a technology. So we did a very rigorous breeding from CMAP, and we have brought a variety which is less latex actually, because this heroin consists, the latex consists of a huge amount of heroin. So what we thought actually, we can reduce the latex production so that we can automatically, we can develop a non-narcotic seed poppy. In North India, the seed is used for cooking purposes, many purposes actually. So this variety named as a Sujata, and this is a very good example for the classical breeding. So now you come to the present status. Now what is the next generation plant breeding? That is, the major one is the gene silencing or RNA interferences. And another one is the genome editing. Many people are using this technology. And this genome editing can accelerate plant breeding because this improved traits without introducing a transgene. The problem in our country, especially the developing country, even in many other countries, the GM crops are not allowed to grow by the farmers. The GM means genetically modified. What is genetically modified? You are introducing a gene from one species to other species. So that type of crops are called genetically modified. But here we are not introducing any gene from any other species. So Government of India is considering to allow this type of CRISPR-Cas edited plant, not under the GM crops. So there is a number of possibilities to do a lot of work in CRISPR-Cas, and introduction of precise modification directly. Suppose you don't want a character, you are seeing a particular gene is major cause for the particular disease. Suppose you don't want actually, because of that the production is reduced. So carefully what scientists are trying to do, remove that particular gene from the whole plant, so that will help to develop a variety without that disease. But it is not so that much easy. Okay, you need a complete genome, you must know the genome, otherwise you cannot edit that plant actually, because you don't know what are the other functions that will directly or not indirectly involved by that particular gene. So n number of issues are there, but still there is a hope for this genome editing. So this CRISPR-Cas, that is a clustered, regularly interspaced, short palindromic repeat. This is a very good system. N number of research we have started, and Government of India we approved this technology. And even government level, national programs are made. Even I think DST, DBT, they will come out with many track proposals, innovating proposals after post-COVID period in this area actually, in the CRISPR-Cas area. Why it is very important? The one advantage, as I told, it is not covered in uh, genetically modified GM crops actually. So some principles Government of India DBT is making. So if it materialized, okay, then this is a very big boom. Okay, so second one, what I want to say to the botany students or scholars or teachers, the in vitro culture of plants. This is very common one. In vitro culture, that's nothing but the tissue culture related work. But only we used till date mainly for the conservation purpose actually. But where we want to take this to a large scale production of secondary metabolites. How it is possible? Suppose we do conservation through the in vitro culture, that is every day we are doing, but how it can be developed into industrial processes. I just gave one example actually. There are n number of plant you cannot cultivate, okay, but they are very important in the point of economical point of view. I have given one example. You just see vanilla. You know vanilla, everyone knows actually. Vanilla that produces 4-hydroxy 3-methoxy benzaldehyde, that is nothing but vanillin. Okay, but what is the problem with vanilla? It is very niche specific. You cannot grow everywhere, and it requires a support of a tree, and initial flowering it takes 3 to 4 years. And very important problem, it requires manual pollination. So because of these restrictions, many people they don't want to cultivate vanilla. So what happens? The natural production of vanillin is completely reduced, and whatever you are using, whatever you are eating, whatever you are smelling or feeling, the aroma of vanilla is nothing but synthetic actually. What is the problem with synthetic? Synthetic doesn't make any difference, but till we have a mindset, it should be from the natural. So there is an alternative species that is called Decalapis hamiltoni. Many Decalapis species, they have the same isomer, that is the 2 hydroxy 4 methoxy benzaldehyde. Without any alteration, it is as like as vanillin. So it has but the plant, the problem with the plant is this is a RET species. Okay, RET, you know, it is a threatened species and it cannot grow in everywhere. But the beauty of this crop actually, this has the vanillin in the root tubers. So what is the advantage? Why don't we use the in vitro method to produce this vanillin through the hairy root culture? So this type of research, n number of Reliance industries, especially Tata industries, they are especially doing this actually. What I want to say, whenever you take a tissue culture project, instead of taking directly a tissue culture project, if you take a part industrial applications, that will be very much helpful for searching a job actually. So now for the in vitro culture just slightly changed from conservation to harvesting important chemical compounds actually. The third one is a very important aspect, plant as biofactories. N number of industries are working in India as well as abroad also. So what we are doing, somebody told the vaccine production is important. How we can apply the vaccine production actually? So once the vaccine come out, is it possible to make it reach such a type of huge country? So that thought they made, and they are making the vaccines through the crops like rabies, herpes, hepatitis B. So when you are eating these crops or plants, automatically that is impounded with many of these compounds. So this is a very wonderful research. N number of works are going on. Plant-made vaccine, antigens, and biopharmaceuticals. This is a one area. If you have slight interest in pharmaceutical and you have a biology background, definitely it is a very good area. I have shown some of these compounds used for uh, human vaccine, and that is through the plants actually, using the potato, maize, spinach, the rice, so that every day we will take all these things and automatically we will be immunized actually. So that is the plant using as a biofactory. So this is one important area. And another important area, plant-based drug discoveries. This is not new actually, but very important. Now they are getting a lot of importance in this era, especially the COVID era, and even the third world countries, the developing countries. We need n number of drugs from the plant-based actually. So a lot of things are there. Artemisia is a very good example, which eradicated the cerebral malaria from the Artemisia annua plant. The artemisinin is the compound and anti-ether is the precursor for that actually, and this got the Nobel Prize. So this type of research, when I say plant-based drug discovery, it's not so easy, that is true, but there is a huge scope for this. I just gave some example. If you take the Taxol, that the Taxol is from the Taxus, it's a very good anti-cancer molecule. Like that camptothecin, again anti-cancer molecule. Like that vincristine, again it is anti-cancer molecule. Disoxylum pagnitriform, this is again anti-cancer molecule. So I have given some example. These all are plant-based anti-cancer drugs. They are in clinical trials actually. So there is a huge area, unexplored area. It's not like that we have explored completely with a huge, vast biodiversity in this country. We have Western Ghats, Eastern Ghats, Himalaya, Andaman Nicobar Islands, Northeast, such a huge, wonderful biodiversity. We have explored, I think, not even less than 5%. So there is what I want to say. This is a area a biology scholars can focus. Slight chemistry knowledge you may require, that's all, and you can go do wonderful research in this area. So even now, latest papers actually, now people started to explore the secondary metabolites from spice and herbs or from medicinal plant for COVID 19. N number of reports are coming. I have read recently one paper. They have reported. You just go through this. These are the metabolite from the plants. They are trying for this for the antiviral compound that is equal to hydroxychloroquine and remdesivir. Even hydroxychloroquine, if you see, that is from the plant tree cinchona. Okay, for some 20 years back, there's a huge cultivation in Darjeeling and other West Bengal area. So n number of research is going on to find out this type of compound from wild resources or the biodiversity, maybe from microbes, maybe from plant, whatever it may be. So what my point is, you just utilize these opportunities. And again, this is very important area, because whenever we say plant authentication, okay, now the subject is moved from plant identification to plant authentication. Okay, earlier people used to say the word plant identification. What is the difference actually? Plant identification stops with taxonomy. Okay, when I spell the word taxonomy, many people may get poor actually. But when you say plant authentication, the plant identification, plant authentication. So this taxonomy, this academic purpose leads into industrial applications. So earlier you used many traditional taxonomy involves morphology, cytology, some macroscopic character. The issue is we need trained personnel. A lot of issues are there actually. But nowadays, a lot of chemical based, HPLC, HPTLC based, even DNA based plant authentication, they have come. That is called nothing but the DNA barcode. So what is the advantage of all those things? They are the safety issues leading to organ failure, this adulterant, potential adulterant. So the it enter into industrial applications. Now, this authentication helps the herbal pharmacovigilance sector in helping to resolve causes of adverse reaction. What are the adverse reaction? There are n number of adulteration going on, knowingly or unknowingly, of course. So what is the major advantages actually? Mislabeling, the n number of mislabeling. The morphologically they are similar, vernacular names similar. So n number of issues are there in adulteration. So there the industry require proper authentication. That's why I'm telling. And I have given some example. Becopha moniri, that is also Brahmi. Centella asiatica, that is also Brahmi. This is a vernacular confused one actually. But if you see the traditional herbal medicine enterprises, many people label that Becopha moniri and use the plant Centella asiatica and doesn't have that particular compound, and which they claim this may be used for that particular clinical purpose. So that leads to organ failure, carcinomas, many things, and allergies, reaction, and long-term organ damage actually. So the Ayurvedic, the Siddha, or any traditional systems are very good, but there always a small issue. What is that issue? Safety issue. So you must be having little cautious about all using this actually. So the industry want authentication. So when the authentication issue is coming, definitely this DNA barcode technology or HPLC technology or HPTLC technology definitely in the scene. So I have given a small example, not only for this actually. Suppose I will give one example. For citrus plant, conventional international trade of endangered species. Suppose Picrorhiza purova, that species is banned for export. So what they used to do, they take license for Androcrapis calve, that is paniculata. After some time, they start to export Picrorhiza. So what happened? You can develop DNA markers for these two species, which can differentiate, because once it become powder, once it become enter into product actually, it is very difficult to identify by your traditional methods actually. So even you can make this type of barcode marker for RET species, as well as generally highly export people, the plants. Another one area I want to tell that's very important area is omics actually, where many people want to enter, especially the biotechnology student. They lot of students, they have n number of interest in this area. So what is the base of this omics technology? that started with a genome sequencing of a model plant Arabidopsis. It's a very recent science, just 10-15 years back. So 2000 it started till date. In 2013, 49 different species full genome sequence has been completed actually. So from this genome sequence or from this omics, what we are going to get actually, you just go through the slide actually. So because we may not know, oh, they are just sequencing the whole genome. What is the use of that actually? It is not like that. From the genome, we can get n number of inbuilt information that may be involved in a trait, that may be in a new secondary metabolite gene, or maybe a predicted gene model, or governing a particular trait. So, etc., etc. Suppose it is a rose actually. That may be a new candidate gene we can isolate or we can identify for recurrent blooming actually. So n number of applications are there that will lead to metabolic engineering, development of genetic marker, or improved breeding strategies. So these omics technology definitely help for the advancement of different sciences, not only your botany. It's a huge area that may help. I just give one small example. I think you may know, all the botanist biology student may know, this is a catharanthus. This is a periwinkle plant. It's a very complicated plant. What is the importance of this plant? A very good important anti-cancer drug, that is a vinblastine, that is derived from this, or ajmaline derived from this plant. But what is the other side? You need 500 kg of dried leaves to produce 1 gram of vinblastine. Just imagine how much it is very difficult. So n number of groups are working this pathways. So what is the importance of the pathway? Unless until you don't know the pathway of a particular compound, how the plant produces a particular compound, suppose methanolide A or vithaferin, we cannot do any work actually. What is the pathway? What are the genes involved in? So these all are covered by the omics technology. So it is very important. You just see the Gloriosa. The Gloriosa pathway, recently they published this paper. Gloriosa is grown very nearby states in Tamil Nadu. More than 3,000 acre it is cultivated. But till date, it is producing the colchicine. I think colchicine, you may know, it is a very good important compound actually. This inhibits the formation of protein filament, that is the microtubules. You can read this paper, very good paper. They have elucidated the biosynthetic pathway of colchicine. So what is the use of this actually? Now, because this is a manual pollinated crop, it is very difficult to pollinate. People pollinate themselves actually. It is very difficult. So once the pathway identified, we can go for which gene is involved in colchicine production. If you find out the gene, definitely you have n number of scope actually. Another area is transcriptome studies. A vast scope in this area. What is the transcriptome study? Just identifying the gene function. So it is very easy. We know some plant is known for some activity, but we don't know exactly from where, which part, and which gene is behind that, one gene or many genes. So to study that, we are doing these transcriptomic studies. Another one important area is synthetic biology. Is a very new area, very important area. All the biological research scholars must read at least 5 papers in synthetic biology. What is synthetic biology? It is nothing. We are asking the microbes to produce the job of the plant system. How it is possible? Suppose there is important compound, very important compound, but you cannot ask the plant to produce like catharanthus. Okay, it is producing very low amount, or it is a rare species. We cannot destroy the species again and again. Or I have shown this plant. It is cannabis. Cannabis. It is very restricted cultivation, very restricted plant. So how we can get the goodness of this plant? Like CBD. CBD is cannabidiol or cannabidone. How we can get the goodness of this, a very good painkiller? How we can get this actually? So there the synthetic biology helps. What we are seeing, the complete pathway we are studying first actually, and identifying the gene actually, and expressing that particular gene in microbe system, maybe a yeast or maybe a bacteria. So that is called synthetic biology. There is a huge scope in this area. So please, scholars, use this. N number of people are entering into this area because it is very lot of scope is there. Industrial scope is there. Job opportunities are there. How? One side, the plant cannot produce that much quantity. Other side, the plant itself rare in the nature. Other side, the compound is so important, and the industry is demanding. So such a type of compound, if you select, if you express it with another system, definitely there are like that. There is another example I have given. Again, this is a yeast expressed actually. So what these are saying, these areas are very important for the biology scholars. So just I want to say again, the latest engineering, agriculture engineering area, n number of artificial intelligence, they are drone controlled, many works are going on. N number of scopes are there in for the, especially the biology scholars. There is endless opportunities. Don't think you don't have opportunity. You have a endless opportunity. It is very important to identify what is very important actually, and where is the gap actually. That is very important. If you identify the gap, then at least 50 percentage or more than 50 percentage you have achieved. And just I will this now we have lot of data. We have to convert the data to solution, and n number of plant tried, so we can solve our problems. And n number of concept, proof of concept we have, that can be converted into technologies and plant derived chemicals. We have now the methodology is there. We know how to go for. Now simply we have to go for the byproduct using the synthetic biology and bioinformatics studies actually. So I will end with this quote. You can go through this quote. The future is definitely unknown, okay, but somewhat it is predictable. So, but look to the future, we must look back upon the past. That is where the seeds of the future were planted. I showed the first slide. What was the major invention? Okay, so the future, to know the predictable unknown future, you first look into the back actually, back upon the past. That is very important. So with this Albert Einstein words, this is the tree. As I told, you never ever imagine how the cotyledon will become such a huge tree actually. So just I want to say, don't ignore, don't underestimate any subject. All the subject, they do their own part and they all are important. With this one, I have completed my first part of the research talk. Any questions, any doubt is there? Then I can go for the second part, career aspects in CSIR. Or Dr. Stephen, shall I continue with the second part?
Yes, sir.
Or any questions I can answer?
Yes, dear participants, if you have any questions so far, you can directly ask to Dr. Sundaresan.
Okay, I think I can go for my second part, right?
Yes, sir, you can continue. They will put it in the chat box if they are having any questions.
If any question, you can read later. Okay.
Sure, sir.
So this career prospects, what I covered in the first set of slides, these are the different science research aspects you can focus. There are n number of scopes are there. But here, what I want to explain is the CSIR, because where we belong actually. CSIR is, you may know, many people may know, exposed actually, you know, JRF exams, everything. That is Council of Scientific and Industrial Research. It is the foremost scientific and industrial research organization in the world. It is established in 1942 and capacity more than 4,000 scientists, 8,000 scientific and technical supporting staff, and 38 state-of-the-art institutes we have actually. And the main objective is any research which will help to the industries. That is the major objective of this actually. So what we have, we are working in 6 different disciplines. That is biological sciences, engineering sciences, information sciences, chemical sciences, leather and environmental sciences, and physical and earth sciences. So these are the 6 major area the CSIR is working. So if you see every field, almost all the field, maybe healthcare, maybe energy, agri-nutrition, biotechnology, ecology, environment, chemicals, including leather, petrochemicals, aerospace, electronics, instrumentation, civil infrastructure, engineering, mining, mineral, metal, material, etc., etc. You name anything, CSIR work actually. So this is a broad classification. We have brought especially the six actually. So among the six actually, so what a biological sciences scholar can enter. So I have given three continuous slide how a biological science students, whether you may a zoology person or you may be a botany person or anything actually, a biology can enter into different aspects. The first one is biology and biotechnology. We have divided into 3-4 sector for the biological science. The first one is the biology and biotechnology. So the thirst area, if you go to the thirst area, it is making biomolecules actually, the research in genomics and biomolecules. The main area is to produce viable product. Viable product may be including through proteomics, protein engineering, drug development, biomarker or nanobiotechnology, etc., etc. So this many labs are involving in this work. Maybe CCMP is working, CMAP is working, Botanical Research Institute is working, IHPT Palampur is working, n number of institutes are working. The bioprospection, that is very, very important aspect. There is a huge scope in this area. Bioprospection is nothing but conversion of biological resources into useful product or patent or technology, etc., etc. So that is very important. But many of the microbiology student, zoology student want to enter in this area. And novel agro technologies, because we know the crop plant likes many environment, but nowadays if you see n number of drought resistant, flood resistant, salt resistant agro technologies are required. So that type of varieties we want to develop. Transgenic crop, as I explained, through the CRISPR-Cas technology, and next generation gene edited plant, and biodiversity conservation. It is again a very important area. The forest biotechnology area, biodiversity conservation is a very important. NBRI, National Botanical Research Institute, is completely focusing on this biodiversity conservation. So if you are a biological science student, if you are interested in biology and biotechnology, you can enter into this test area. Why I am saying this actually, once you completing the process of selecting a particular thesis title, then you are entering into your work actually. Don't do that. Instead of that, go through such a type of organization's website. What is their test area? What is their plan for next 5 years? And what is the government plan, or where government want to focus actually? So if you select all those things, it is very easy to choose your problem. So just not for the sake of PhD. Okay, it is not good. It will add a number in your guide's biodata. That's all what you will get actually. So instead of that, go through the websites, go through the different aspect in these labs actually. And again, the another one is, if you go the same biological science scholars in healthcare is another important part, and government of India is focusing on that. Government of India want people should enter research on this healthcare actually, stem cell research, especially many top focused diseases like diabetic or parasitic diseases. Now COVID-19 entered actually. So we have again the scope added. So n number of scope is there compared to genomics, biology. So there is a vast scope in this area. Psoriasis, again, very important part. Tuberculosis, arthritis, diabetes. So these are some of the healthcare sector where government want to invest money. So why don't we take research on these areas? Again, another one is agro and food industry. It is very, very important area. Agro and food, CFTRI like institutes are working here. Here, the main focus is food technology, genetically modified food. So here also n number of biological sciences scholars are required. And again, ecology and environment. As I told, biodiversity, natural resources, NERI is working, NBRI is working, IHBT is working. So a lot of research programs are going on to site. You can do your research locally. No need to go to the institute and carry out the research. You can do in St. Joseph College, or you can do your own college, universities. But take the problem, the national problem. That is very important. How to find out the national problem? go through these websites. We have time to time update what is the national program, what are the test areas for the next 5 years, etc., etc. So how we can enter into CSIR? It is very easy. It is not a very big tough task actually. First, if you want to do PhD, you must clear UGC net actually, CSIR net or UGC net. So net with JRF, net without JRF, there are a lot of advantages, disadvantages. If you have net, you can choose your institute and you can choose your guide. So if you have net, we are running one program that is ACSIR, that is called Academy of Scientific and Innovation Research. This is the ACSIR PhD program exclusively run by CSIR, but only you can do the research within CSIR only. So what is the strength? We have 2000 full-time faculty and 3000 students enrolled. Already 2 batches completed actually. So through this ACSIR, you can complete your PhD if you have any net, any fellowship. So fellowship is must. If you don't have fellowship, at least you must be part of any project. So you will be called as a project assistant. And another one is lot of fellowships are available. You can avail Diamond Jubilee Research Intern Award time to time. All the 38 laboratories, the website, time to time, they display all these things. The tenure is 2 years, the eligibility is master degree. And Dr. Shyama Pratash Mukherjee, SPM Fellowship, I think MSc student, all you may know. So that is the master degree, top ranking in JRF actually, net. And senior research fellowship. If you completed 2 years your research and you have good publication, definitely 2 years experience, one publication, you can enter. This is for 3 years, SRF actually. And research associate, you have a PhD, but not time submitted or awarded, but not much time is over. You can apply for that actually. And Nehru Science Postdoctoral Fellowship, this is introduced very recently. And senior research associateship. So these are some of the opportunities awarded by CSIR. And recruitment, you may not know how they recruit. Time to time they advertise in the website as well as that one. In generally, you have to check the website, biology institutes, as I told, like NBRA, CMAP, IHPT, n number of CCMP. Okay, so junior scientist to chief scientist till they will recruit actually. Lateral entry is also permissible in CSIR, and technical officers, research scientists, and there are n number of schemes. CSIR research grant, travel grant, partial financial assistance. You want to go for attending your, presenting your paper, etc. N number of schemes are there. And fellowship for scientists, there is a prestigious Raman Research Fellowship they award to expose in foreign countries. Even you retired emeritus scientist scheme is there. And n number of awards are there. Santi Swarup Bhatnagar, that is the Science Nobel Prize. They used to call the Indian Nobel Prize for Science actually. So we have Santi Swarup Bhatnagar Award, CSIR Bhatnagar Fellowship, n number actually. So the scopes, there's n number of scopes are there. You can approach any CSIR laboratory. We will ready to guide. Thank you very much. Dr. Stephen?
Yes, sir. So participants, if you have any questions, you can directly post it in the chat box, or you can directly ask Dr. Sundaresan. He is right now available.
Anything posted in the thing?
So far, not yet, sir. So far, there's no questions.
Okay. I think I have completed in time because it is almost
Yes,
the climate also. I think everybody is in house. We are in office.
yes, sir. Sir, I have one query. Especially doing PhD at CSIR institutes.
Yeah.
Is there any other prerequirements the students should have, or they can directly approach?
No, there are two things actually. One is you are entering with your fellowship. Okay.
Yes.
You are coming with your fellowship, CSIR, JRF, or NET, or anything, any fellowship, GATE, any fellowship you are coming. DST Inspire, many DST Inspire fellows are joining. You have no restriction. You can straight away, you can select your laboratory, select your own guide also, and you can send a mail. You can meet him personally. You can talk, interact. What is the subject you want to do? You can enroll. There is no problem. And enrollment, there are two PhD programs are running in institutes. One is ACSIR, another one is Jawaharlal Nehru University, JNU program. Okay, especially in SEMA Bangalore Center, we have applied for the recognition as a research center to Bangalore City University, BCU. Okay, once we got the recognition, I think many of the students can do wherever they want, they can register in BCU, right? And another one mode, they can time to time we call for the projects actually, for our gap projects, sponsored project, or aroma mission type of project, mission project, 5-year plan project. So they can enroll in that project as a project assistant, but it takes 1 year to complete, at least one paper is required. Then they can enroll for their PhD. This is the mode actually. Other than this, there is no other mode. One with fellowship, one without fellowship, but a stipend with a project actually. Okay. But registering through these two one is JNU, another one is the ACSIR. Okay.
Sir?
Yes, ma'am. Beatrice, ma'am, go ahead, ma'am.
Sir, I just wanted to find out, like in Bangalore, like in the ACCSIR, supposing you register, I mean, the guides are already affiliated to Bangalore University or to any other university since it's a CSIR laboratory?
ACSIR, we are running our own course. Okay.
Yeah, so you give the PhD degree or?
Yeah, we give the PhD degree. You are under the ACSIR because it is head office is in Delhi, like your UGC PhD, okay, or Jawaharlal Nehru JNU PhD, right?
Correct.
Now what we have applied to Bangalore City University, right, for the guide recognition. Okay. If it is awarded, actually we are awaiting for the approval. Okay, because our jurisdiction, it is coming under Yelaganka. Okay.
Correct.
So we can register. We are already some 7-8 scientists are there and dealing with almost all subjects, starting from botany, biotechnology, microbiology, etc., etc. Okay.
Yes.
So once it approved, then there is no need of enrolling CSI, ACSIR or JNU. Then it is a Bangalore City University rules actually.
Okay.
They have to attend the coursework, everything here, and they can attend the lab here. Got it?
Okay. Okay. Thank you, sir.
Yes, dear participants, you can unmute yourself and you can ask directly.
So I have a question. Sorry, this is Dr. Vajan from Microbiology Department.
Yes, sir. Go ahead, sir.
I also want to ask if there are any facilities available for students for their master's dissertation. PG microbiology or botany students, they do their dissertations. So are there any facilities available for the same?
Yeah, very good clarification. Okay, very good clarification. In fact, I told many university faculties also, you must make it as a compulsory the last semester as a 6 months project actually. Okay. The project 6 months project courses, we used to call it as a GT, graduate trainee. Okay. It is available, but not less than 6 months. Okay. What we observed last 2-3 years, when we take free of cost, okay, because we have to spend a lot of money on them. So we fixed fees for them for 6 months. Okay. It's a 25,000 rupees fixed fees actually. Okay. If it is less than 6 months, it is 15,000 rupees, 15. Okay. Why the fees? We have to spend a lot of money on them. And what is the guarantee? They definitely joined with a project, ongoing project. And when they complete, they must have one internal guide from their department, right?
Yeah.
Their department, and we guide from here, and they have to spend whole time here actually. Attendance will be here. There is no okay, but every month they have to go to their department and they can present their updates actually. What is their report? Okay, and Viva is conducted by the department. We don't have anything actually. Finally, the thesis will be awarded from your department only. We give one certificate, but what is the benefit? There will be a combined publications. Okay, definitely the scholar and the internal guide actually. Okay, so there will be a combined publication. So the scholar will get a publication when she enters into a PhD itself. That will be the advantage, sir. And they will be exposed into high technology. And we don't run like a training course. Okay, within 15 days they will be trained. They have to handle everything themselves. So how many of the colleges are doing, universities are doing, I will tell you, they sent a bright student. Suppose Dr. Stephen is there, okay. He has a very good student. That student also want to enter into research area. Got my point, sir? So what he will send 6 months training to our lab, and that candidate will a particular area. Suppose Dr. Stephen wants to enter into synthetic biology, okay. So he tells a student, go and get trained. Okay, synthetic biology 6 months, and we all will get a publication also. After 6 months, the student will go and join with Dr. Stephen and establish a laboratory. Actually, that much training we give them. Actually. Got it, no, sir?
Okay. Yeah. Yes, sir. Yes, sir. Thank you. Sir, I also have one more question, because our MSc microbiology, they have this dissertation 4th semester. They normally go to ISC, NCBS. They already are doing this. Okay. So we thought we'll explore your this one CSR also. There are also some students who might not be able to, pay. So is there any sort of, concession or scholarship that can be availed or something for those students who are economically, weaker?
We don't have such a type of thing actually, because these all decision taken by centrally. Okay, it is not one institute, right?
Okay.
If in that case, what we used to do, you can enter into a collaboration mode actually. Got it, na? Suppose you have a project, you have a project work, so both institute can do in a collaborative mode actually. In that case, when collaborative mode, we cannot charge money actually, right? That is possible, right?
Thank you, sir. So whom do we contact for such things, like, whom do we
have
write?
Dr. Dinesh Nagegoda. We have a scientist in charge. Okay, you can write to them. Website it is given. cmap.rs.in is a regional center. Or if you are in Bangalore, definitely you must bring your students actually one day. Exposure visit, because people are from Kerala, Tamil Nadu, Andhra. They are coming actually. Okay. See, IASC, NCBS, they are very big institute. I am not saying, okay, but they must be a public partnership institute like CMAP. They will learn many things. Entrepreneurship they learn, societal work they learn. Okay, not only a science. We don't want to make them only a researcher. As Dr. Marimuthu told, there is a part societal research. You can do n number of work in that area, sir. So that exposure they will get only from CMAP. That I can assure.
Sure. Right? Thank you. So one last question. So how is the current situation? COVID, are you taking students now for
Yes. We are, sir. The day from the day I think lockdown, we are working, because government asked all the scientists should work. Okay. There is no restriction now. I think almost all opened. I think except academic institute, research institutes are working, sir. There is no holiday. Nothing.
Okay. Thank you, sir. Thank you for your answers. Thank you.
Sir?
Yes.
Will you be taking students at least now, if they are willing to come?
Yeah, any time is okay. The only thing is it should be at least 5 to 6 months. Otherwise, there's no use. Many people approach for summer, 1 month, 4 weeks. That won't help actually. They cannot also learn, and that is not useful for us also. No. Okay, if they committed for 5 to 6 months, then definitely they will involve in their work. Got my point, no, madam?
Correct.
Yeah.
So I just wanted to know, like, since they do almost like, what if they do it like for 6 months, but they have to give their thesis within 4 months, but they will continue after that?
4 months, actually, not the thesis work should be completed. It is not like we always we always give some seriousness for the work. Okay.
Correct.
So within at least minimum 6 months required for a good thesis to complete just isolating something and that protein isolator, that is thesis formality will be completed, but that won't give you a publication. Actually, our target is at least one publication, madam.
True,
if they get a publication, you can see the student point of view. I told in many, even registrar, even many BOS also, make it 6 months. They can attend the classes in between. Suppose they have Monday they have classes, okay, or Saturday they have classes. They can attend some research methodology classes, but they must spend at least 5-6 months, madam.
Okay, thank you, sir.
That is my
thank
thought.
you, sir.
Sir, one Sundaresan, sir,
I saw one question to work on omics technologies.
On omic technologies.
Yeah.
We have to know the whole sequence or partial sequencing is
at least draft sequence is important. Okay, without complete ignorance we cannot do anything actually. Okay, because many of the project we are receiving, even DBT, DST, a plant they want to work in genome editing, they want to develop a seed bearing plant from the vegetative, many things are coming. But when we saw the publication, there is not even a draft genome actually. So definitely partial genome is important actually. If it is full length, it will be very good, well and good actually. Okay, you can target very easily. Anything?
So I have one more question. Can I ask? So,
Yeah,
yes,
please,
sir.
please.
Yes, sir. Go ahead, sir.
yeah, this is for faculty. Are there any sort of workshops or conducted based on the techniques and all of that, instrumentation and all of that for
Yeah,
a
there are
month or
many,
thing
many,
during?
many queries are coming, sir. We are planning actually. Okay. But the issue is we have to conduct only for the accommodation is the issue. We don't have accommodation, but many people want to conduct. Okay, we have recently entered into a MOU with the Karnataka State Science and Technology Council. Okay,
Okay.
so I think future we will conduct with them, right? Because we have very good facilities, there is no problem, especially for the faculties, right? We have plans, sir. Definitely let you know. Actually, definitely let you know. Okay.
Sir,
Thank you, sir. Thank you.
Yeah.
can you hold something locally for the local teachers at least, and then later on you can think of a,
Yeah, that
I know.
we received actually. How we can conduct for locally? That is the main issue, because many people are asking. Many queries we have received, especially from other states. Actually, I think from our state it is very less people, because we have a lot of institute in our city. Okay, people can go JNCSR, people can go IHR, people can go IASC, people can go NCBS, right? We have opportunities are more. We have a lot of options. I can say options. Okay, but we planned 2 3 times, but it couldn't take. I think we are a little serious. We recently conducted one online international conference. I don't know how many of you got a chance to attend. It was an international conference, 3 days online conference. Okay, it was a very good it's not like just a formality conference. It was really serious actually, very good talks, very eminent invited people from it was a metabolic engineering. It was exactly in metabolic engineering. Okay, we can plan actually. Okay, in sometimes some colleges or some universities came for a focused work practical. Suppose that is possible, very easy. Suppose you have a suppose you have a small subject, a chapter, you have a practical actually complete
Yes, sir.
sequencing. Okay, so we can make a 7 days program for your class actually. You can come, you can start from DNA isolation, standardization, PCR, and learn for 1 week practical. Instead of your laboratory, you are conducting some 7 days practical in our laboratory. Got it, na, madam?
Yeah.
Okay.
Will there be any charges, sir?
No, that in that we don't have any charges.
Okay. Thank you,
because
sir.
that is purely, it is a type of
Teaching.
academic service actually.
Yeah. Thank you so much, sir.
Yeah, thank you.
Dear participants, if you have any questions, you can directly ask. It's almost time. I think,
Okay, yeah.
yes. I think there is no more questions.
Yeah.
So thank you so much, sir, for your thoughtful lecture on recent trends in plant science and career opportunities in CSIR. You have taken us from classical to modern techniques of plant or molecular biology. You have highlighted that what are all the opportunities available in biological sciences, and especially in CSIR, what are all the opportunities we have. Especially, it will be useful for the students and the early career researchers. On behalf of St. Joseph's Research Center at St. Joseph's College, I would like to thank the resource persons, both Dr. Sundaresan as well as Professor Marimuthu, for their wonderful time and their thoughtful lectures on biological sciences, especially research methodology which will be, I hope it will be useful for all the researchers, especially uh, the participants from India as well as abroad. We have quite a number of participants from abroad, so it will be a very useful. And already in the chat box, we are receiving a lot of feedbacks. It is a nice presentation, nice session, and excellent. All these things. So on behalf of St. Joseph's Research Center as well as on my own behalf, I thank Dr. Sundaresan as well as Professor Marimuthu for their wonderful time, and they shared their valuable knowledge with us. Thank you so much, sir. And
Thank
also
you.
I thank management, especially for the principal as well as for the director, research director, for this wonderful opportunity given for us everyone to listen a wonderful lecture, especially all the managements from the St. Joseph's College. We thank everyone and all the participants who participated actively, both interactively, they've participated. I thank each and every one of you for from the bottom of my heart for your patience listening as well as interactive mode of thing with a resource person. Thank you so much. Have a wonderful evening. Be safe during this COVID time. Thank you so much.
Thank you, Doctor.
Thank you so much, sir.
Okay.
Participants can fill their feedback form. Once you fill the feedback form, you can leave the meeting. Thank you so much.